Students, today we are going to see some of the diseases affecting the mustard crop, which is also another important oilseed crop. And the mustard cultivation is more prominent in northern part of India compared to southern part of India. And this mustard is susceptible to many diseases such as Alternaria blight caused by Alternaria brassicae, and then white rust caused by Albugo candida, renamed as Cystopus candidus, and then downy mildew caused by Paranospora parasitica, and then powdery mildew caused by Erysipeae cruciferarum, club root caused by Plasmophora brassicae. Club root we would have studied only in the case of cabbage, where it causes finger and toe symptom or club root symptom on the roots of cabbage plant. Similar pathogen is also causing club root symptom in mustard crop. And then Sclerotinia stem rot caused by Sclerotinia sclerotiarum. Here also the Sclerotinia pathogen is known to produce white head rot in the case of cabbage. By covering entire cabbage leads to rotting and mycelial growth. And similar to that of cabbage, here also the bacterial blight or black rot, which is caused by Xanthomonas campestis or campestis, which is also a serious issue. And some of the minor diseases like soft rot caused by the bacterial pathogen Yersinia cartovora, and then mosaic disease caused by cauliflower mosaic virus, and then rye mosaic virus, which is causing turnip yellow mosaic virus causing mosaic disease, that is caused by cauliflower mosaic virus as well as turnip yellow mosaic virus. And then first one is the Alternaria blight, which is otherwise called as black spot or grey spot, which is caused by Alternaria brassicae, which initially produces small circular brown necrotic spots with concentric circles and coalesce together to form patches of infected area and blighting appearance. And when the disease is severe, it covers the entire leaf and leads to defoliation. Circular to linear dark brown lesions may also be formed on the stems as well as on the pod, and which elongated at the later stage. The infected parts, they may produce small, some discolored or shriveled seeds. This is the typical spores produced by the Alternaria brassicae. So the pathogen that survives both externally as well as internally and seed borne in nature. So from the infected seeds, it is primarily spread to in the faster rate, and the pathogen may also survive for a long period on the infected plant debris as well as the weed host. So the relative humidity of more than 75%, and then the warm temperature of 15 to 25 degrees Celsius with the intermittent rainfall, which are favoring the disease development. So if the disease is very severe, as I told you, the infected plant debris may serve as a source of inoculum, then we need to go for crop rotation and then remove the infected seeds and use only the healthy seeds which are showing high germination percentage. During the cropping period, if the incidence occurs, then go for spraying with the copper oxychloride or mancozeb at the rate of 0.1%, or even go with the application of biocontrol agents like Trichoderma viridi, Trichoderma harzianum, and Bacillus subtilis, etc. And some of the varieties which are showing resistance against this disease are reported, which includes Safal, Agrani, and Bina Sarisha 3, Bina Sarisha 4, Bina Sarisha 5, 6, 7, 8, etc. And then the white rust, which is caused by Albugo candida, which is recently renamed as Cystopus candidus. So this pathogen, white rust pathogen Albugo candida, which is known to cause both local infection as well as systemic infection on the plants. The local infection that includes production of white creamy yellow raised pustules on the undersurface of the leaf, especially when the disease is severe. You may see also on the upper surface, and later they coalesce together and form a large patches on the surface of the leaf. The systemic infection and the humid weather, which causes some mixed infection of white rust as well as downy mildew symptoms, which causes swelling and distortion of stem as well as floral parts, which is due to the hypertrophy and hyperplasia of the phenomena, hypertrophy and hyperplasia phenomena, and it produces stag head like symptoms, which are all brown, hard, and dry as they become mature. And this is the typical stag head symptom due to the infection, which shows hypertrophied and hyperplasia cells of the stem, leaf, as well as the important floral pods. The albugo is known to produce subepidermal sporangiophore and sporangia. The sporangiophores are short and stout, which bears chain of oval shaped sporangia arranged in a basipetal succession, and every sporangia get attached with each other by a specialized structure called isthmus or disjunctor, which is made up of gelatin. And the pathogen during the sexual reproduction stage, it known to produce oospores as the sexual spores. So the primary spread is mainly from the oospores present in the infected plant debris as well as in the soil, and secondary spread may be due to sporangia, in turn the zoospores. So relative humidity of more than 70%, and then warm or low temperature between 12 to 25 degrees Celsius, and intermittent rainfall favors the development of disease, and some of the cultivars reported as a resistant, which includes Argentine, Basanti, Maya, that is RK9902, and then Javagar mustard 2, Him Sarsen 1, which is mentioned as ONK1. And if the disease is very severe, then better go with the crop rotation with the non-host crop for minimum period of 3 years, which may be helpful in to avoid the further occurrence of the disease. And then the next important disease is downy mildew caused by Paranospora parasitica. So during the initial stage, it affects the seedlings, and discolored lesions may be developed on the cotyledons later that turn into necrotic. Upon the older leaves, small, angular, light green or yellow patches on the upper surface of the leaves, and grey, white masses of spores or downy growth produced on the undersurface of the leaf. The lesions get enlarged and the development of with the development of disease, and form irregularly shaped necrotic patches on severely infected leaves. The most conspicuous and pronounced symptom is the infection of inflorescence, where it is known to produce hypertrophy of peduncle and as well as inflorescence, which also develop stag head structure. So while studying about the albugo disease in the previous slides, we told the albugo is presence of albugo is also associated with the downy mildew symptom. So both combined together, they may produce hypertrophy and hyperplasia of inflorescence, petiole, peduncle, etc., and produce stag head like symptoms. And the pathogen, downy mildew pathogen, is known to produce hyaline, sinusitic, highly branched mycelium, and the sporangiophore are long and dichotomously branched, and these branches are at acute angle. This Peronospora is known to produce sporangium on equal branches, and during the sexual reproduction, and this sporangia in turn contains numerous sporangiospores or zoospores, and during sexual reproduction the pathogen produces oospores, and the oospores, which are double walled, thick walled, dark brown in color, which remain viable for a long time in the soil as well as in the infected plant debris, which serves as an initial source of inoculum. And then the airborne sporangia or zoospores, which helpful in the secondary spread of the disease. The very low temperature of 10 to 20 degrees Celsius and the relative humidity of more than 90% and intermittent rainfall or continuous rainfall and leaf wetness, all these factors are favoring the development of disease. So better control the broad-leaved weeds which serves as the which harbors the pathogen, and then use completely the disease-free clean seeds to avoid the development of the disease. And while before sowing, it should be treated with the apron or metalaxyl at the rate of 6 gram per kg. And during the cropping period, if the disease is noticed, then spray metalaxyl at the rate of 0.2%. And some of the resistant varieties like Uttara or Him Sarsan 1 may also be used for cultivation in when wherever the disease is very severe. And the next important one is powdery mildew, which is caused by Erysipelas cruciferum. And this disease is caused by oidium type of pathogen and produces symptoms on the upper surface of the leaf as dirty white circular flowery patches, and later it may cover both the surface of the leaves. Under the favorable conditions, the pathogen produces this type of powdery growth on entire leaf, stem, and young flushes, floral parts, and even upon the pods. It produces powdery growth. And when the disease is very severe, it covers the entire leaf, and the complete leaf turn into dry and leads to defoliation. In the dried and fallen leaves, the pathogen is known to produce the sexual fruiting body, that is ascocarp, sexual ascocarp called Chasmothecia, which in turn contains numerous ascus and ascospores. So the Chasmothecium present in this is the typical Chasmothecium, which is having a myceloid appendages, and here the conidia are produced from the conidiophore, and the conidiophore which arises from the mycelia, and the conidiophore are short, stout, and which bears a chain of barrel-shaped conidia arranged in a basipetal succession. The Chasmothecium, sexual ascocarp, or the Chasmothecium, which is present in the infected plant debris that serves as primary source of inoculum. Chasmothecium, in turn, the ascus and ascospores. So these ascospores are the initial inoculum, and secondary spread is mainly through the airborne, barrel-shaped, single-celled hyaline conidia. This is the barrel-shaped, single-celled hyaline conidia. And the disease is favored by the warm, humid weather, and warm, humid weather and low or no rainfall period, disease will be more severe. When the disease is more severe, then go for crop rotation or remove the debris which serves as initial source of inoculum. And then some of the varieties reported as resistant. These two, that is Uttara and Himsarsan 1, also reported to be resistant to both downy mildew as well as powdery mildew. And then avoid application of excessive nitrogenous fertilizers should also be followed. When the disease is appeared during the flowering season or upon initiation of the disease, then go for spraying with wettable sulfur at the rate of 2.5 gram per liter of water, or Carathane or even carbendazim at the rate of 0.1% were found to be effective in reducing the disease incidence under field level. And then next one is the club root rot. The club root, earlier we have studied in cabbage, where the club root is favored by the acidic soil, so the infection that leads to development of root like swollen knob like clubs or nodes on the infected roots and rootlets. So the disease is caused by the protozoa, that is Plasmodiophora brassicae. The affected plants remain stunted. Why? Because there is no absorption and translocation of water and nutrients, and hence the affected plants may remain stunted. Tiny nodules, tiny size to larger size, club shaped outgrowth with develop on the roots as well as rootlets, which completely prevents the absorption and translocation of water and nutrients. Ultimately, the upper portion, the leaf, which shows pale green and yellow color, and which is showing typical wilting at a later stage. Under severe infection, the entire plant become dead due to poor translocation and maintenance. The pathogen remain survive or remain active in the soil as well as in the infected plant debris as resting spores. So initial inoculum is from the resting spores, and humid weather, high soil moisture, and acidic nature of the soil all are favoring the development of this disease. And so to get away from this incidents, you could use certified pure disease free seeds. And then as I told you, the acidic condition of the soil favors the development of the disease, and hence apply lime to the soil in order to reduce the in order to bring up the pH and reduce the development of pathogen. And the next one is sclerotinia stem rot or white mold caused by Sclerotinia sclerotiarum. Initially, the pathogen produces elongated water-soaked lesions on the stem at collar region or near the soil level or at the crown region. Later, it produces profuse white mycelial growth, premature ripening, as well as shredding of infected stem, and then wilting and drying of entire plants are the associated symptoms. And in severe case of infection, upon the affected stem or portions, it produces brown to black sclerotial bodies, and later the infected plant may dry and completely dead. The pathogen survives as mycelium on the dead as well as the live plants, and it also known to produce irregular black sclerotia on the infected plant parts, and this sclerotia present in the soil for a longer time, or it may present as a contaminant along with the seed. So the initial infection is mainly from the sclerotia present in the soil, which germinates and causes that water soaked elongated lesions at the collar region of the grown up plant. And high humidity of 90 to 95% and an average temperature of 18 to 25 degrees Celsius along with the wind current are favoring the development of this disease. And to manage this disease, go for crop rotation. That is the best choice, as the pathogen is known to produce more of soil-borne inoculum, that is sclerotia. Better go with the So for managing this disease, go with the crop rotation. As we discussed earlier, this soil-borne sclerotia will be much more, and which serves as the initial source of inoculum. Use complete, clean, healthy seeds and avoid dense population of the plant. And this Hims sarsen 1 earlier, which is also told to be resistant to downy mildew as well as powdery mildew, that is also showing some of the resistant reaction to this pathogen. And during the cropping period, if the disease is noticed at the early stage itself, go with the spraying of azoxystrobin at the rate of 0.1%. And the next important disease is a bacterial disease, which is caused by Xanthomonas campestris, Pathovar campestris, which causes black rot symptom. Similar type of disease is also reported from cabbage. There also it is mentioned as black rot caused by Xanthomonas campestris, Pathovar campestris. So the infected leaf tissues turn yellow and show chlorosis, and the disease progress towards the center of the leaf in a V-shaped pattern, and the veins are also show brown or black discoloration and causes vein necrosis. Dark colored streaks are also formed on the stem from the ground level and gradually enlarges, causes girdling of the stem. Girdling means the symptom encircles the entire stem and causes the plant above part of the plant just above the point of infection got dried, and the stem become hollow due to internal rotting. The midrib show cracking of, especially in the lower leaves, and browning of veins, withering of leaves are also associated symptoms. In severe cases, the vascular bundles of the stem turn into brown and the entire plant will collapse. The pathogen, that is bacteria, which survives in the infected plant debris as well as in the seed, and the pathogen secondary spread is mainly through the rain splashes or irrigation water. Warm and humid climate weather which favors the development of disease, and as I told you, the pathogen is soil borne and it remain active on the infected plant debris, and you have to go for sanitation, field sanitation. And then if the disease is very severe, go with the non-cruciferous, go with the crop rotation with the non-cruciferous crops for every 2 years, and then spraying of streptomycin sulfate at the rate of 0.05% would be effective, but this application of antibiotic should be applied with care, because development of antibiotic resistance in the pathogen is very severe phenomena, and you cannot control the pathogen at the later stage. And alternatively, the same streptomycin sulfate is also been used to control some of the diseases in human beings. So when we consume it is also indirectly may develop resistance against streptomycin sulfate in human being also, whenever they consume such infected heads or cabbage crops. And then cauliflower mosaic virus, so which causes cauliflower mosaic disease on the mustard. So it produces a typical chlorotic mosaic mottling pattern and mosaic banding on the infected leaves. It also causes vein clearing symptom as you see in the picture B, and ultimately leads to malformation of leaves, rugosity and stunted growth of the plants. And later stage, the affected portion, it produces necrotic spots and complete yellowing of leaves, malformation of leaves, and vein thickening are also prominent in the affected plants, and which is caused by cauliflower mosaic virus, which is a double stranded DNA virus got transmitted by the aphid vector Myces persicae in a semi persistent and non circulative manner. So in order to reduce the incidence, better go for the vector control. Metacystoxis, spraying of metacystoxis at the rate of 0.1% is effective against the sucking pest, or any other effective pesticides against the sucking pest can be used to reduce the vector population. Thank you.